All right, welcome back to part two of translation. By the end of this lesson, I'm hoping you will be able to describe the four levels of protein structure and compare and contrast the functions of proteins. So where we left off last time is we had the formation of a peptide bond between a three amino acid long polypeptide chain and a new amino acid coming in. Um, and if we take a look at this from a graphical standpoint, we have this first amino acid. It is methionine. You can see that it has a free amino group, so it is the amino terminus end. And then we have the second amino acid. Here is its side chain. Here is its amino group and its carboxylic acid group. You can see that both ends are in peptide bonds. So then valine and tyrosine and cysteine. Cysteine is the last of this group, or the last amino acid of this chain. And you can see it has a free carboxylic acid end, or a carboxyl terminus, or the C terminus. And you can see here how methionine is coming from the AUG codon, serine from the AGC, and so on and so forth. So there are 20 standard amino acids that can be found in polypeptides. We did introduce you to a 21st and a 22nd. Briefly, we may go into that a little bit more later. But let's just talk about the 20 standard amino acids. And again, these are universal across all organisms. Each amino acid has a unique side chain, or what we call an R group. Again, this should be largely review. We can break up those side chains into different groups that have particular chemical properties. So this first group here is what we're going to call the nonpolar group. We can also call it the aliphatic amino acids. But these all have side chains that are nonpolar in nature. So here, just a plain hydrogen, here, just a methyl, here, kind of a branched chain here. Here, it's the only difference is it's one carbon further out. Here we have 4 carbons, so 1, 2, 3, 4 in leucine. This is isoleucine, 1, 2, 3, 4. So it still has 4 carbons, but they are slightly rearranged. Again, different structure, different function. But all of these are more or less nonpolar in nature. These last two, cysteine and methionine, and this first one, glycine, are kind of barely nonpolar, but for the most part, the bulk of these tend to act in a nonpolar way. All right, here we have another group. This is a group that we can call the aromatic amino acids. These are also nonpolar in nature in that they are hydrophobic. But these have a particular unusual ring structure present in them that has double bonds. Double bonds, again, are pi bonds, and they can do some unique things that these others cannot do. So we'll put them in a slightly another subcategory of hydrophobic. So now we have just the aliphatic amino acids and the aromatic amino acids, both of which are nonpolar in nature. All of these in a protein that is, or a polypeptide chain that is in an aqueous environment, are going to tend to be, or it is going to be folded in a way that all of these amino acids are wanting to point into the interior of the protein. So just the um, forces of hydrophobicity is going to push all of those in together so that we can minimize the number of water cages. And this is the bulk of what begins the protein folding process. Next, we have the polar and charged amino acids. These are all hydrophilic in nature. They are going to tend to be on the surface of a protein because since they can form hydrogen bonds with water, water will tend to prevent them from being folded into the interior. There are, of course, always exceptions to that, to both hydrophobicity and hydrophilic. All right, so this first group here we will call the polar amino acids because they are polar in nature, but they are not charged. So here we have serine with just an OH. Here we have threonine, which has an OH in a slightly different position. It's essentially a methylated version of serine. Here we have asparagine and glutamine, very closely related. They have a carbonyl and an amino group, both kind of unique in nature. Down here we have polar but charged amino acids. In this case, these are acidic. So these all have carboxylic acid, and you see them here in the ionized state. So the hydrogen proton has been ionized off, leaving its electrons with the oxygen, giving it a net negative charge. And here we again have a slightly longer version of that. Over here, we have 3 amino acids that are basic in nature, meaning that they have a tendency of absorbing protons. So these release protons, making them an acid. These absorb protons, making them a base. These are all shown in their protonated form, so they have already gained that proton, and that gives them a net positive charge. Here we can see histidine, which has a unique ring. Um, Here we have lysine and arginine. Uh, Histidine is additionally unique in that it can become charged and uncharged fairly easily. And there are also circumstances in which this ring structure can take on a net negative charge. So the versatility of this amino acid means that it tends to show up in particular sites, such as the catalytic site of enzymes. All right, here are those 2 oddball non-standard amino acids. Here is selenocysteine, and this is essentially the same as cysteine, except for it has selenium instead of sulfur. And if you look at a periodic table, you'll recognize that this is the next element down on the periodic table. So its chemical properties are very similar to sulfur, and sulfur is actually the next element directly down from oxygen. So all 3 of those have similar binding characteristics, but different electronegativities. And here is lysine. So it has a kind of a lysine core right here. You can see that reminiscent over here. And then it has this group attached onto the end. All right, so there are 4 levels of structure in proteins. Again, hopefully this is a review. We have primary, secondary, tertiary, and quaternary. Quaternary is not present on all proteins. So the protein's primary structure is just simply its amino acid sequence. This is what is being dictated by the nucleic acid sequence. That's what I meant to say here, my apologies. So this primary structure is completely dictated by the genetic code. And that is all we mean by the primary structure. So again, the primary structure of this unit right here would be methionine, serine, valine, tyrosine, cysteine. Just that sequence. All right, those sequences have unique chemical natures, and that is going to cause the backbone to fold in order to hide hydrophobics and keep hydrophilics exposed to water. There are particular shapes that tend to form repeating patterns. These repeating patterns are called an alpha helix and a beta sheet. Now, there are particular amino acids which are good candidates for alpha helices or for beta sheets, and there are particular amino acids that cannot exist in alpha helices or beta sheets. So the amino acid sequence is certainly going to help dictate this. The unique characteristics of alpha helices and beta sheets with hydrophobic and hydrophilic environments is certainly going to play into it. But what is happening here is we have this structure that is stabilized by formations of hydrogen bonds between the atoms located in the polypeptide backbone. So it's backbone-backbone interactions, hydrogen bonds, that are stabilizing these two particular structures. So here is an example of an alpha helix, and here is an example of a beta sheet. So here for the alpha helix, this is the N-terminal end, that is the C-terminal end. Notice that there are these green segments sticking out. That is the position where we would find the side chain. So in an alpha helix, all of the side chains are in fact pointed outwards. On the interior here, you can see that there is a hydrogen bond that forms between the carbonyl of one part of the peptide bond and the amino group in a peptide bond several amino acids up. And that pattern repeats over and over and over again. And this is a particularly stabilized structure. Here we have a beta sheet. You can see here that the beta sheet is essentially a fully extended version of a polypeptide. And a single aspect of that is called a beta strand. When you have two beta strands that are next to each other, they form a beta sheet because they can again form hydrogen bonds between the backbones. Unique characteristic of a beta sheet is if you consider it to be a flat piece of paper, every other amino acid will point towards you, and the next amino acid along the path will be facing away from you. So here we have facing away, facing towards us, facing away, facing towards us, facing away, facing towards us. So both of these things you can kind of predict when they might occur from the primary sequence. If you see over here every other amino acid being hydrophobic and the alternate every other one is hydrophilic, that would be a clear indicator that that's probably forming a beta sheet. Over here, it's every 3 or 4th amino acid that shows up, or side chain that shows up on the same side of the cylinder. So if you see every 3rd or 4th amino acid being something hydrophobic and the rest of them hydrophilic, well, that's suggesting that it may form some type of alpha helix. Beta strands can occur in both parallel or anti-parallel configuration. So in this top panel, we're showing anti-parallel configuration with the first strand's N-terminus on the right, C-terminus on the left. The next strand has its N-terminus on the left, C-terminus on the right. Down here, we have both N-termini on the right-hand side. One thing that you might notice is that the hydrogen bonds seem a little bit more uniform in an anti-parallel arrangement than they do in a parallel arrangement. The result of this is that parallel arranged sheets tend to have a more extreme curvature to them, to where anti-parallel sheets have a slightly flatter appearance. Um, Parallel sheets can be so extreme in this curvature that it can actually be formed into a barrel shape, which we do see in some proteins. So here I'm just showing examples of both an alpha helix, and next we'll show a beta sheet. Again, just showing you how the side chains are arranged in comparison to the backbone. So here we're looking down the core of an alpha helix, or down the barrel, so to speak, and you can see how all of the side chains stick out. In this figure, the side chains, green is carbon, red is oxygen, blue is nitrogen, so you can tend to see that this side is polar and this side is nonpolar in nature. Over here, you can see again that every other side chain is sticking in the opposite direction. And if you consider this to be the flat sheet, then you can see how this definitely creates some sidedness to the sheets. Again, reinforcing the idea that seeing particular patterns in amino acids suggest particular secondary structure arrangements. Now, the reason why these secondary structures happen has a lot to do with this peptide bond. Notice here we have a carbon double bonded to an oxygen, and then bonded to a nitrogen, and that nitrogen is bonded to a hydrogen. On either side, you can see what we call the alpha carbon of the amino acid. So for amino acid number 1 here, here's its alpha carbon. It is the central point that contains both the side chain as well as the amino group and the carboxylic acid group. Over here, we can see the carbon, the alpha carbon of the following amino acid. The presence of these two electronegative elements in this configuration has a unique effect, and it is because the electrons that are existing in a single or as lone pairs that aren't involved in bonding can cause the double bond to flip back and forth between here and here. So what happens is this double bond, or these lone electron pairs, comes down, forms a double bond with the carbon, and that boots out one of these electron pairs that is in a double bond, and it turns them into a lone pair, and that can revert back. Since we have a double bond that is sometimes present here, sometimes present here, it tends to fix the bond angle between this carbon and nitrogen, allowing no rotation to occur in between those two bonds. This whole thing kind of creates a flat planar structure with the oxygen, carbon, nitrogen, and hydrogen all existing in a single plane. The reason why these can move around is because they are existing in a double bond, which occurs in a pi orbital. I'll leave chemistry to come in and talk about that, or I can touch on it briefly in class. The end result of this is when you have a particular amino acid and you have its alpha carbon, you can get rotation around the bond before and after, but you do not get rotation around the peptide bond, okay, on either side. The fact that we have such limited flexibility allows us to kind of name these particular bond rotations. So again, we'll go to the Greek alphabet, and we'll call this the phi angle, and we'll call this the psi angle. And there are particular angles at which rotation results in a collision. Namely, as you turn it, you get into a conformation where these two oxygens try to uh, exist in the same space. This, of course, cannot occur, but it does serve as a useful reference point. So if that angle were to occur, we refer to that as 0-degree rotation psi and zero rotation phi. If you start looking at all the different possible configurations of phi and psi, some of them are forbidden because of similar activities or just simply because they're in a unusually high energy state. And we can plot that on a graph. So here we have a graph that has the phi angles on the bottom, the psi angles on the y-axis, and the 0-point rotation is right here in the middle. So right here at the crosshairs is that collision point between the two oxygens. Again, something that is not possible to exist. If you look at all the dots on the graph, all the dots represent structures that are in the database where you see particular bond angle pairings. So you see particular phi-psi angles existing in a protein. You can see that there are a lot here, a lot here, a lot here, few over here, few over here and over here, but you get large regions of this graph that are completely unoccupied. This is occurring, again, for two reasons. Some of these are just not allowed, such as this crosshair region. There are some other regions that are not particularly allowed either. But then there are particular regions that not only are allowed, but are stabilized by the formation of hydrogen bonds in these secondary structures. So here we have all the beta sheets existing over here, the alpha helices existing over here. And these create low energy states, which make them favorable. One thing that you'll notice is there are more things listed on this chart. There's this thing called a polypeptide 2 helix and a 310 helix. This goes to demonstrate that although we talk about beta sheets a lot and we talk about alpha helices a lot, they are not the only secondary structures. They just are the most frequent. If I were to give a third most frequent, I would say it would be this 310 helix. So what is a 310 helix? Well, here's an alpha helix, here's a 310 helix. It's essentially wound slightly tighter. And you can see this has a unique effect on where the side chains are. They all tend to line up on one side. But you can see here that this is part of a spectrum. So we actually have this thing over here called a pi helix, which is wound looser. We have something in between a 310 and an alpha called an omega helix. So all these configurations are possible. We have this thing called a polypeptide 2 helix. This tends only to occur in proteins that have a high proline content. Because of that, they often call it the polyproline 2 helix. And this is because of the unique nature of proline. The fact that its side chain actually comes back and is bound to its amino group. This is an important structure because it is what exists in the protein collagen, and you may have heard of people getting collagen infections before. This is a major filament type structure that is part of our connective tissue. It's also a major part of our bone as well. There's also these things called beta turns. These are particular arrangements that we see frequently. They do have self-stabilizing hydrogen bonds. And you do see particular amino acids at particular positions. Glycines usually tend to be involved in here, and sometimes you have prolines involved in these turns. So there are more secondary structures, and again, these secondary structures are low energy states because of limitations on the backbone rotation, and also the formation of hydrogen bonds make them low energy. All right, So again, we had the primary structure of a protein, that's simply the amino acid sequence. We talked about how there are all these secondary structures, and we went into great detail on that. Now let's talk about tertiary structure. Okay, and tertiary structure is just simply when we take these secondary structures and fold them up on themselves and create a three-dimensional structure. This is largely determined by hydrophobic interactions and then ultimately stabilized by ionic hydrogen bond and van der Waal forces. But hydrophobic is what tends to force it into a particular conformation. So if you have this unfolded noodle of a protein, here hydrophobic is marked as yellow, blue is more hydrophilic. So when we have this existing in an aqueous environment, water is trying to form cages around all of these hydrophobic structures. It's much easier just to force it to hide all of those hydrophobic structures in the center. So when you take this drive of hiding the hydrophobics, combine it with the limited flexibility of the backbone, allowing it only to occur in particular conformations, and the fact that secondary structures may have formed along this path, you end up getting a three-dimensional structure. And so here's another example of that. So here we have a floppy noodle. Here we have some secondary structures forming spontaneously, and it's folding up to hide all those hydrophobics, and now we have a defined three-dimensional shape. Here you'll notice there are alpha helices, there are beta sheets, but we also get these loop regions. These loops may be actual flexible noodle-like structures that can flop around and interact with the aqueous environment around it, or it may be something more rigid. So if I were to look at this structure here, I would say this loop and maybe this loop down here might be somewhat flexible, but this little loop here and this loop right here, which has a lot of interaction with the rest of the protein, is probably not flexible. So just bear that in mind. So again, we come back to the idea of tertiary structure, but there is a level beyond that that we call quaternary structure, and this is reserved for when we have more than one polypeptide chains existing in an overall protein complex. So here to put this along a schematic, primary structure is all dependent on its amino acid sequence. This is driven by the DNA and is covalently bound. Secondary structure is caused due to limitations in the flexibility of the backbone and also interactions occurring forming these hydrogen bond secondary structures. These come together and with the hydrophobic forces forcing it into a tertiary structure, and this is the kind of final shape. This may or may not interact with other proteins to form a larger complex, which we'd refer to as a quaternary structure. Now, we also have these things out there called motifs and domains. Motifs are simply small secondary structure arrangements or combinations that we see over and over again. So here's a coiled motif. We see it occur very frequently. This is a helix loop helix motif. We see it happen over and over again. So we've given these their own names because of that. Here's something called a zinc finger. You can see it's called that because it binds zinc in it. You can see this one over here binds calcium. So these secondary structures have common patterns to them, secondary structure and patterns, but they also tend to have some type of functionality involved with it. So here the coiled motif is a very common way to bind two proteins together. You might have this section of a protein binding the two polypeptides chains together, and then on the end of each one, you might have a separate area that does something different. The idea of having a separate area that has a separate function is where we get into the idea of domains. So oftentimes proteins will have particular parts of the protein that serves a particular function, and then a whole nother part of the protein that serves a different function. So here's an example of that. Here we have something called the EGF precursor protein. This is what is produced in order to make this thing called EGF. This is also known as epidermal growth factor. That is something that, as you might infer, is a growth factor. It binds to cells, it signals them to do something. Specifically in this case, it's a trigger to start replicating, or one of the triggers. It's not the only one that's needed. And as the name implies, it has to do with the integument system, or the skin. But you can see here that there are these other proteins as well. So here we produce this protein as a big precursor with lots of copies of this individual domain. It is then cleaved off into these individual pieces. But here we have a whole nother protein that just so happens to have an EGF domain embedded in it. Here we have something else called tissue plasminogen activator, and you can see here that it has that domain, but there are also these other domains as well. What this does is it allows us to link particular functions of a particular domain with the functions of other domains. So here, this is a free-floating entity meant to diffuse and interact with other cells. These proteins over here are meant to both bind the same protein that binds binds EGF, namely the EGF receptor, but it also has other functionalities as well. So maybe it has a kinase and can phosphorylate and activate something. Maybe it has a binding site for another receptor, and clustering the two receptors together is an activating signal. So we have this idea of domains and we have the idea of motifs. Both of them seem to have some aspect of function to it. The main difference is that motifs are solely little patterns, and they aren't their own thing. They always exist in the context of some larger protein. So there's something called calmodulin that has two of these, but then it also has a hinge region in the middle that binds other proteins. As I said here, this is oftentimes what you see with proteins that are forming dimers. They have this region here strictly for the formation of dimers. The rest of the protein, the part that actually does work, is down on one end or the other. So motifs don't tend to have a full protein structure stability aspect to it. It's just little minor repeated patterns that we see, to where domains, you could separate those domains from each other and they would still retain their entire structure. So whatever this pink oval is, if you were to trim it right here, it would still maintain its shape. It may not have this, and it certainly wouldn't have the same function being separated from the other domains, but it would still maintain its three-dimensional shape, just as EGF retains its three-dimensional shape when it's produced as a series of multiple and then cleaved off into individual chunks. So there is a whole protein aspect to this domain idea. And what you'll notice is if you look at the evolutionary history of particular proteins, in scenarios like we have here with this TPA protein, if you look in bacterial cells or lower organisms, what you might see is several proteins that actually form a multi-protein complex that serves the same function as this overall protein. What higher organisms have done is just cut to the chase and connected them all together into one long polypeptide that allows its function to be achieved easier. Recognizing that back in evolutionary history, this may have had a solely separate aspect in bacteria than it does in yeast, and very different than what we may have. So just to give you an idea of how domains work. So you might consider motifs and domains falling in between the secondary structure and third and tertiary structure. They're kind of somewhere in that spectrum, but they have unique names and have unique usages for those terms. We oftentimes represent proteins in many different ways. So here is your ball and stick figure. This is what you would happen if you took one of those chemistry modeling kits and tried to actually build a protein out of it. You can tell here that it's very complex. It's very hard to see what things do, but it does give you an idea of what the three-dimensional overall shape of this might be. Here we have something called a alpha carbon backbone trace. So here we just have the backbone traced out, and from that you can see where alpha helices are. You can kind of see where there are beta sheets existing, but you just see how the protein is wrapped in on itself. Here we have something called a ribbon diagram. It's essentially a backbone trace, but we've replaced the alpha carbon and the beta sheets with these cartoon representations. And this just simply allows us to see the secondary structures more easily. So we can see that there is a parallel beta sheet over here, and that it is bound on one side to this alpha helix, and you can see that this whole thing is sitting on top of another beta sheet, something that isn't as easy to see up here. Here we have something called a water accessible surface. So we've essentially taken the electron clouds of all these and created a surface to this. Notice here that although this looks like there is space in between here and here, it doesn't take into account its full spectrum of the space that the electron clouds take up. So when you actually look at it in this conformation, you can really see what the surface of the protein actually looks like. Here we have a stick figure of a molecule that is binding into the surface of this. And you might recognize this here as ADP being bound into the surface. Here we've colored it based upon its electronegative qualities. So again, nitrogen is blue, but is also representing basic amino acids. Red is representing acidic amino acids. White is neutral in nature. Here we have a hybrid of the two. This will show the surface, but also shows the substructure underneath by making it semi-transparent. Now, proteins have a lot of different functions. They are the thing that makes cells cells. They do all the various things. We usually think about proteins in terms of enzymes being things that catalyze particular reactions, but these are also things that are giving shell its shape. Remember, the cell membrane, the phospholipid bilayer, is just oil. It is not something that has structure to it. It's the cytoskeleton underneath that gives it its structure. But this is proteins can also be involved in movement. So it's proteins interacting that cause muscles to contract. We can have cell signaling events. Cell-to-cell interactions might be facilitated by this. We have transport proteins, so we have sodium channels in our cells. We have hemoglobin, which transports oxygens inside of red blood cells. So there's all the various jobs that you see in a cell are mostly done by proteins. So there is quite a diversity in this. I would encourage you to also go back and review the Bio 101 videos, namely that in macromolecules and focusing in on proteins, particularly taking a look at the amino acids. I may yet again talk to you in class about those amino acids, about how individual amino acids may have particular functions, like histidine and proline, what makes them special and unique. So I would encourage you to review some of that material as well.